We have about 10 to 40 billion capillaries per body, and capillaries are typically very easily accessible. So if you prick your finger, that's usually what bleeds. Capillaries are usually in 1 millimeter of most of our tissues. And that makes sense, right? To facilitate the diffusion of oxygen and nutrients. The total surface area of capillaries is about 600 meters squared. So we have a lot more capillaries than we do the other branches of the vasculature. And that is what's responsible for the slowing of the flow or the decrease in the velocity that we see. Capillaries are about a 1 millimeter long, and we have a small length here, but we do need some length again to facilitate that diffusion, okay? In between our endothelial cells, we have some pores, and these pores facilitate the movement of the materials that we're going to be speaking about, as well as the movement of protein-free plasma. Really important to point out that proteins cannot cross capillaries, not easily, at least not the capillaries that we're going to be speaking mostly about. Neither can cells, right? So cells are going to stay behind, and all that's going to move is the watery component of blood, which is the plasma or the fluid. We're going to be speaking in the context of capillary beds. So we'll look at the structure of a single capillary, but understand that the network of controlling flow happens in terms of a capillary bed. So arterioles lead to capillary beds, and so we can regulate based upon that level at the bed. So this is illustrating that concept of slowing down of flow. So the graph on top is showing the cross-sectional area against the distance throughout the vasculature. So as we move through the aorta, the arteries, and the arterioles, we see a smaller cross-sectional area, and we see it significantly higher at the level of capillaries. And think about an analogy of a river that is emptying or dumping into a lake. So because we have many more capillaries than we do arteries and arterioles, this is where we get that slowing down of the velocity. So the graph at the bottom shows that slowing down. So once again, we had a higher velocity in the aorta, and by the time we get to the capillaries, we have a significantly slower velocity of flow. Okay. Now there are three types of capillaries, two main types that we'll speak about. And the most important one, so if you don't remember any of these, remember the continuous capillary. That is pretty much the one that we're going to be speaking in context of when we look at diffusion and when we look at the movement of materials in between cells and tissues. So the continuous capillary is the most abundant, and there are very small gaps in between the endothelial cells, which facilitate the movement of, once again, that protein-free plasma, as well as some water-soluble molecules to go from the blood into the tissue and vice versa. There are also fenestrated capillaries, which have slightly larger gaps in between the endothelial cells. And this forms fenestrations or pores or windows. And this allows slightly bigger molecules to fit through. So we have proteins fitting through. In some cases, we can even have cells moving through, okay? There's also discontinuous capillaries, and this is the branch that connects fenestrated, sort of a transition between fenestrated capillaries where we have small pores to sinusoids. Sinusoids have really large pores, no basement membrane, and this is basically a free flow or a free exchange of materials. And there are certain areas in the body where that is really convenient, okay? All right, so let's illustrate that here. So we got continuous, and this is the most abundant type. We're going to be speaking mostly about this one. There's fenestrated, and then there is discontinuous, which eventually leads to sinusoids. So we'll draw the cross-sectional view of these 3 capillaries. And we'll draw our endothelial cells. Which are lining these capillaries. In fenestrated capillaries, we mentioned that we have much larger openings in between these endothelial cells. We call these fenestrations or windows. And then in discontinuous capillaries, we have even larger spaces and we have no basement membrane. So this is pretty much a free flow or free exchange. So in continuous capillaries, we have a narrow endothelial cell gap. Can you guys read my handwriting? It's an embarrassing question. Okay. Okay, so now our endothelial cell gap, and this facilitates the movement of mostly fluid, but also water-soluble molecules. Okay. Now we're gonna have slow flow or slow movement of molecules here. Simply because we have smaller pores that these molecules have to move through. And then we also know that this is the most abundant, so I'll make a note here. In fenestrated capillaries, we have wider endothelial cell gaps. These are called fenestrations. This allows for rapid flow or rapid movement of molecules. And then we can also see some larger molecules getting through here. So larger molecules like proteins or cells can kind of squeeze through fenestrations. Some of the examples in the body are going to be in the kidney, where we have reabsorption and secretion. We also see this in the gut. We also have reabsorption and secretion there. And then in the endocrine system, where those hormones must make their way into the blood. Okay. And then finally, discontinuous sinusoids. This is a transition basically from fenestrated to sinusoids. And sinusoids are basically blood-filled sacs. They're blood-filled sacs in the body. Here we have the free flow of materials. Examples in the body are going to be in the liver. This is where we make proteins like albumin, which are much larger. They have to get into the blood. We also see this in the bone marrow, where fresh red blood cells have to make their way into the circulation. And then we also see this in the spleen, where blood cells are recirculated or recycled. Okay, so these are the 3